I used to visit all the time. I've been busy like out of the way. Hi everyone, good evening. How are you guys doing? We're gonna go ahead and get started. A little bit behind schedule. All righty. Welcome everybody to our November program. It's an interesting thing. Usually we have this November program before Thanksgiving, but now we're doing it after. I even went back a few years on the calendar to make sure, do we usually have this before Thanksgiving? And yeah, for the past few years it has been, but I hope everybody had a great Thanksgiving. You know, this is for me one of my favorite time of year, and it's a great opportunity to give thanks for all the things that we have. One of the things being the camaraderie that we all feel in these meetings with like-minded folks who all have a common appreciation for plants. So just wanted to express my own gratitude to be able to be in this position and get to see all you guys all the time and share in that passion for native plants with you. There's a lot of thanks to give, for all you guys, for being members, for being participants, for being volunteers, for being native landscapers, for contributing in any way you can in promoting native plants and their importance. And thanks, of course, to a lot of folks from the board. This doesn't happen without them. All the volunteers that we receive, and it's truly a collaborative effort. So. The last big thank you I wanted to give was to everybody who donated recently at Give Miami Day. We made an incredible close to $3,000 from 38 donors, and that's all that goes into. Yeah, round of applause. For everyone who donated, truly, it's very much appreciated. All those funds go towards allowing us to continue the same programming that we do and offering the field trips and continuing to try and educate the community at large about native plants and native ecosystems and why we should value them, as well as promoting and acting as an avenue for folks to acquire some of those native plants. So as always, we have a few announcements before our main event. So first and foremost, on Sunday, December 3rd, there will be the Miami Blue chapter of NABA. They will be having their quarterly meeting. There was supposed to be one earlier this month, but there were some technical difficulties. So they're going to have a follow-up on December 3rd. There should be some information in the back table if you're interested. The topic's going to be at-risk butterfly conservation and recovery efforts in Florida and beyond. Of course, we all know there's a very intimate link between our native butterflies and our native plants. Can't have one without the other. On Saturday, December 9th, there will be a really cool field trip to Fakahatchee Strands Preserve State Park. It's going to be from 8:30 to be determined. Newsletter says sometime after lunch. Knowing us botanists, folks, lunch is going to be at like 3:30, so out there a good length of time. It's going to be led by orchid enthusiast and state chapter member Russell Plusman. It is a member-exclusive field trip, and it's limited to 20 participants. So if you are interested in going on the field trip, contact Steve Woodmansey at steve@pronative.com ASAP, because these spots they tend to fill pretty quickly. There's more information on the newsletter and website calendar. You know, some of the logistics, you know, things to bring, etc. On Saturday, December 16th, there will be a chapter workday at Everglades National Park at the Ernest F. Coe Center. If you are a new volunteer and you're interested in participating, please contact Patty Fairs at fairspl@gmail.com. Tuesday, January 23rd is our next monthly meeting. The title of it is Foraging Florida, Finding, Identifying, and Preparing Edible and Medicinal Wild Foods in Florida. It's a new book by Roger Hammers. He's going to be talking all about it. And I'm sure also selling the book here too if you're interested in acquiring it. On Friday, March 1st is the application deadline for the FMPS Science Award. So these are grants that cover a multitude of different areas. So if you have a project that you'd like to apply for, just send us an email, an email application. There should be a template on the main FMPS page. You can just send it to datefmps@gmail.com. Let us know that you're interested, and we'll review. But there's a bit of time. So March 1st is both a world away and also right around. As is Native Plant Day, which will be held this year on Saturday, March 16th. So if you haven't already, definitely mark your calendars. I'd like to take a moment to welcome our new members. So please give a warm welcome to Everglades Nursery, who's joining us as a business member. Lori Prepsent, Olivia Walker, Amy Cuadra, Marcy Ziv, Dorothy Parker, and Justine Derez. Are any of those folks here today? Show of hands. Hi, you'd like to introduce yourself? Oh, it's Oliver. Finally, I look at these things so quick, sometimes I make an error. Pleasure to meet you, Oliver. Glad to have you here. Anybody else? If I made another typo, please let me know. Well, welcome. So thank you to everybody who donated plants to the raffle table. You know, these is always my favorite part, seeing all the really cool plants that find their way here. Just a bit of housekeeping. Kindly turn off your cell phones as our presenters today are giving the presentation. And as always, there's more information on the newsletter. There should be a sign-up sheet if you're not a member to receive the January newsletter in the back. And then last thing, I was informed of a petition that we have some information in the back in regards to clean water. You know, very important thing, part and parcel with healthy ecosystems. So if you're interested in getting some more information, there's a flyer in the back with a QR code as well as the petition itself if you're interested. So that's it for us. If you guys want to keep in touch with us, we have all our social media except what was formerly known as Twitter. So for our online viewers who are joining us via YouTube, we're going to take a few moments to talk about the raffle table plants, and Raul Moas has kindly agreed to present.
Thank you. Okay, so starting here, we have an unusual plant for the raffle. This is Ornodia littoralis, the golden creeper, which is a seaside shrub, grows to about 2, maybe 3 feet, a sprawling, very sprawling plant, pretty white flowers, like dull yellow, maybe golden berries. Very handsome, will get bushy, get a bushy plant. The Jamaica caper, one of the slowest growing of the tropical hardwoods, but one of the prettiest, has a very, very pretty pyramidal growth structure, dark shiny leaves. The capers are actually like the capers that we eat and are edible, I believe. A very nice Simpson stopper, typical forest, hardwood hammock, and also sometimes in the pinelands. Very handsome plant. Food, the berries are very preferred food for birds. 2 Passiflora suberosus, the passion flower that is the passion vine. Host plant of 3 butterflies, the zebra, the Julia, and the Gulf fritillary. And then there are sedges that are sort of alike, but not in habitat. This is Abelgardia, and this is a pineland sedge. It is a clump sedge, as most of them are. Very, very handsome. Will get much bigger than this, actually, if you leave it alone, and will spread fairly easily in good habitat, in open habitat. And this is the Canada spike rush. Is a wetland plant. If you have like a marshy garden or a wet garden or like a water feature, it's nice to go around it. Very rapidly spreading. This is the wild alamanda, which is a beautiful, beautiful, maybe inch and a half yellow alamanda type flower. This becomes a big vine and can overpower small trees, but it is very, very attractive. Skippers love this flower for some reason. 3 white stoppers, Eugenia axillaris. It says it has a distinctive fragrance in the hammock. Yes, it's true. Some people don't like it, like the smell, but it's a very, very handsome stopper. All of the stoppers are fairly slow growing, but it becomes a pretty tree. Lime, it is sources for the giant swallowtail, and I believe also the Schausses may use this. It is a kind of a sprawl. It's wicked, like 1/4 inch recurve thorns. Wicked. So if you're able to trim it back, that's good, but just be careful. It is a great attractant. You will see the giant swallowtail around it. This is a senna. It's listed as Mexican senna, which would be, I guess, Senna mexicana chapmani, but the leaves look more like Senna ligustrina. I'm not sure. If someone has a better sense of this, speak up. I'm not sure. This is Asclepias granata, swamp milkweed. There's 2 of them here. Unlike its name, it will grow in moist, not boggy soil. You don't let it dry out badly. This is probably appreciated and underused flowering. Great butterfly draw. The flowers are beautiful, big heads. Will grow to about 3 feet, maybe 4 tall, perennial. The only drawback is that because it has abundant leaf surface, the monarchs will eat it. So for those of you who don't like monarchs eating your milkweeds down to the ground, there is something called Bacillus thuringiensis. I think the brand name is Thuricide. It is to caterpillars only, not to butterflies, bugs, or any other wildlife. You can use it maybe once or twice. Works fine. This is Simmons aster. 1/4 or 1/3 of an inch wide flowers, not as big as the other wild pineland asters, more of a wet soil, do okay in dry ground. The nice thing about it is lots of flowers, and it will, unlike most of our other asters, creep rhizomatously, so it will spread. Here is a rockland morning glory, Ipomea tenuissima, 3/4 inch pink violet flowers with a deeper purple throat. Very pretty, said to be a perennial in the literature. In my yard, they behave more like annuals, but they easily seed. You can grow them really easy from seed. You just have to get the seeds from after they flower. This is Jacomontia pentanthus, the morning glory. Found, I think, nowadays only in the Everglades. It is sort of like an ecotone plant. You see it around the and pine lands. Beautiful, beautiful. Again, maybe 1/2 inch flowers, but lots of them. If they bloom, they bloom. And it will be a vigorous grower. Lancement volucrata, wild sage. It is probably one of the shrubs that you want if you want a lot of butterflies. So this is a draw to almost every butterfly I can imagine. They will all the skippers, everything will come to it. This is mine. American pokeweed. Those of you who are in the Deep South know that folks in the Deep South eat this. It is toxic. It has to be prepared specially, otherwise you get poisoned. It is sort of like a weed type disturbed soil plant, but it is a great bird attractant. The birds will jump on the seeds, and a lot of bugs eat the leaves. You can see once they get big, they'll be all eaten. So whatever eats the leaves is probably feeding something that eats them. So trimmed back, looks like it had a crew cut, is a Trichum adnatus. This is a scale leaf aster that will get to be a dome-like, maybe about 2-3 feet wide, 1.5 foot tall, wiry, very small leaf plant, but covered nowadays. If you see one in full bloom, it is spectacular. Flowers are about 3/4 of an inch wide, anywhere from pink, blue, light to like lavender color, very pretty. It's like a wiry dome full of flowers. This is Randia aculeata, the white indigo berry. Small tree, large shrub. Some of them have spines. This variety that I have here does have spines occasionally. Pretty flowers, pretty white flowers, attractive to butterflies. The berries are white on the outside, but dark blue on the inside. You squish them, and your fingers turn blue. 5 beautiful tetrazygias. They're really beautiful. These are very nice looking tetrazygias. The West Indian lilac, pineland plant, very common in the pinelands of the Everglades. And then 1, 2, 3, 4 rare ferns that I shan't name because they're named here, but they're all either endangered or extinct in the wild here in Dade County. They're grown by Jennifer and Daniela. There you are. You brought them, right? They're beautiful. So thanks to you and to Jennifer, we will thank her in person. They're spectacular. A lot of ferns in Dade County that are now either gone or almost gone just from habitat loss and development. This is a Buraria tomentosa, the rough, strong bark. And if I am not mistaken, this is a small tree, large shrub. And it has beautiful red berries that I believe is very attractive to birds as well. This is also a rare plant here. This is balloon vine, Cardiospermum. And it is a relatively aggressive vine, but it is the host plant for one sphinx moth at least, I think 2, and several other lepidopterans, including other moths. So it's a fantastic ecological plant if you like to have like moths and such. And that's it.
Cool. Much appreciated. And one more note for the balloon vine. It's also the host plant for the Miami blue. So it's one of 3 known host plants. I planted it a few years ago at home, just one little individual. And they do come back, but they do die back annually too. So you only have them for part of the year. It's a really, really cool plant. All right. So tonight's main event. Usually I'm just introducing folks, just reading off the newsletter, but this one I think deserves a personal touch. Andy's one of the coolest people that I know in Miami. I first met Andy when I was relatively fresh out of college. I'm born and raised here in Miami. I'm a Hialeah, and Andy teaches at Jose Marti MAST in Hialeah. I studied out of state, but when I came back, within a few months of coming back, I met Andy and I saw all the work that he does at Jose Marti with his students. And I saw his botany setup, all the cool things he was doing with orchid germination, with the plants he was growing in their nursery outdoors, the project with NASA, just all the coolest things that make you wish, dang, can I go back a few years and be a high schooler again so I can transfer from Matter Academy, go to Jose Marti for a while and take just that one class. Truly, it's a lot of really awesome work that Andy and his students do. You inject so much passion and enthusiasm for your classes, and you see all the incredible outcomes. You want to go to a prime birding spot in Hialeah, and that's a sentence no one has ever said ever. because Hialeah is not known for that. All you have to do is go to the Arboretum at Jose Marti MAST and see all the native plants that are there, attracting species that I never even knew existed until Andy showed me. So that's all I've got to say. I'd like to give Andy the opportunity to now introduce his students who will be giving the presentation. You can just hold.
Well, thank you very much for that rousing introduction there. But yes, I'm Andy. I was born and raised in South Florida. Raised in South Miami Heights. I used to play in the Pine Rocklands. That was the blimp base to us before Metro Zoo and to Miami, before the prison was built down there, before Larry and Benny Thompson. So back then I was ignorant, though. I played in, I'm sure I destroyed trees and plants that were very rare. But, you know, with age comes a little bit of wisdom. And so when I came to Jose Marti MAST 13 years ago, had the opportunity in an administration that was very willing to let me take risks and kind of have combined native botany with statistics and mathematics and research. And I think I'll agree with you. We've got something cool going on there. Jose Marti MAST is one of the top 100 schools in the nation by U.S. News and World Report for the past 5 or 6 years. So we're doing some cool things there. And I have with me tonight a couple of very cool students. Originally, we had 5 students who helped create this presentation. But circumstances as they occur, you know, prevented 3 of the students from attending tonight. But with me tonight, I have Alejandro Ganasa, an 11th grader, and Nestor Suarez, a 12th grader. So we're going to talk to you today about germination of Tetrazygia bicolor. Just a little bit of background, I suppose. We've been collaborating with the Fairchild Conservation Team for a number of years now with Brian and Jen and Daniela and Lydia to try to give my statistics students meaningful questions that we can help answer. You know, I could go ahead and, you know, have them do an experiment to see what grows mold better, white bread or wheat bread, but there's no purpose to that. You know, with what we have going on with native plants at school, it's very natural for us to go ahead and ask Brian, ask Jen, ask Danielle, ask Lydia, you know, what questions do you have? And the question that Brian had 2 years ago was, and I remember you were kind of, Brian can make rocks grow, you know, and that's how good he is. But you had a bit of disgust with Tetrazygia because you were having a hard time propagating them, so much so that you had the one plant that you got into a 1-gallon container. And you said, here, Andy, just take it. And so the next year, Daniela harvested a number of seeds, and we turned it into our experimental context for the 2022-2023 school year. And that's what we're going to present to you tonight. Okay. So Alejandro and Nestor.
So I think we can get started. Good evening, everyone. How's everyone doing tonight? Good. Oh, a lively crowd. I know. I like present to a younger generation. They'll be like, good. So, well, I'm Alejandro Ganasa. I'm a junior at Jose Marti MAST 6 through 12 Academy.
And I'm Nestor Suarez. I'm a senior at the same school.
Unfortunately. And today we will be presenting kind of a germination outlook on the Tetrazygia bicolor, also commonly referred to as Myconia bicolor these days. But we'll get into that. And so like we said, 5 of our students, well, of our teammates worked on this, and I wanted to say Nicholas Baez, Celine Hutchins, and Chris Miranda Jr., they worked so hard on this. We had several groups do different of the substrate experiments that you're going to see later on, and we all worked in different groups. So it was nice coming together and seeing all the combined data and seeing how much of an effect we actually had together. And all the pictures you're going to see in the back during the presentation are the beautiful flowers that Tetrazygia bicolor propagates with the the white petals and the yellow petals. So, click it. Oh, because I did that. Okay. So what is Tetrazygia bicolor and why is it important? And I think so Tetrazygia bicolor, also commonly referred to as Myconia bicolor, we don't know when the genus name switch was, but we kindly refer to it as Tetrazygia just for the familiarity of it. I think it works better in presenting. And yeah, so the Tetrazygia bicolor in itself is a native shrub. It is found in the pine rockland areas of the Everglades. And I know that's not much these days, but we do find it interesting that it's such a lively plant once you get to actually germinate and see how it grows. So it ranges between 3 to 9 meters. These reach around 10 to 20 centimeters. It has a moderate growth rate. They propagated really fast, so I would say it's a very fast growth rate. And they're found in the pine rockland areas, which are rocky grounds. They have limestone kind of substrate to it, which kind of leads into a hint as to what we actually experimented with. And then we have moist soil type and medium drought tolerance because Everglades is pretty good for that. And then according to the Institute of Regional Conservancy, their status is native to South Florida, but it is imperiled. And the reason for that is Pine Rocklands used to be 185,000 acres of Miami-Dade County, but by 1996, it was only 2%. And imagine 1996 was how many years ago? So by now, it's way less than 2%. And it kind of shows the reduction and how that implements and affects Tetrazygia bicolor in itself, and not just Florida, but in South Florida, but the entire state of Florida. So as an imperiled species in South Florida, we try to assist, and I know Brian came to Mr. Kearns and he was like, I need this, and we need to find a way to germinate this as most efficiently as possible, whether that be through how fast it grows and also their germination rates in the larger scale. So we have 2 essential research questions solely because out of the 4 experimental groups, they kind of shift ways. One being the substrate groups that we use to actually propagate and germinate the seeds. And so it goes back to what type of substrate is the best use. And our 3 experimentals are a clay mixture compared to seed starting soil, a 50-50, well, they're all 50-50, a limestone mixture compared to a seed starting, and our compost mix compared to our seed starting. So they weren't compared to each other, but rather to just a control to see what the actual effects were. And of course, the response, we had both a categorical and a numerical data. So qualitative-wise, we had germination proportions, see how many of the seeds in each cell in each tray were actually germinated. And then from the numerical, it's how many days did it take to germinate. Any questions so far? The second question is for our sinkers and floaters. So I don't know if you see in the picture the little green, well, they aren't green, they're white, but the seeds that are floating. And so our experimental group was those that float and those that sink to see whether or not that kind of had an effect on what the germination rate was. And again, the categorical being the proportions of what we saw, how much was growing and how many days it took to grow. So from here, I'm gonna, one of these parts were presented by one of our teammates, but since this was done, extracting and depulping was done by our botany class, and we were the AP statistics class, Mr. Kearns kind of has the first-hand insight as to what went on during depulping.
Okay, so I was at a workshop at Terra, and I got a bag of fruit, tetrazygia fruit, and that Daniela had harvested, and it was really, really ripe, and it was something that we needed to get to extract the seeds from very quickly. Had 3 AP statistics classes, 2 botany classes. Let's have them work together. So my botany classes, we spent 3 days depulping the seeds. And the process of depulping was this. You can see the materials that we used here. There's a wire mesh, a kind of a coarse wire mesh strainer. There is a beaker, and if any of you had kidney stones, you'll recognize the fine strainer on the right-hand side. Hey, there's multi-purpose things here, right? Here at the center, these are pictures of the berries. And on the right, this is 2 of my students that were depulping these seeds, essentially pushing the seeds through that coarse wire mesh. And when they went through the coarse wire mesh, the seeds are so small that they went into the mixture. We poured water over it to try to get it to come through. So we see that on the left now, that we have 2 students that are now pouring that looks like grape juice, but pouring this mixture that has seeds that are on the bottom that have sunk, and then seeds on the top that are floating. Poured them off into the fine strainer, and then we collected those floating seeds, dried them separately, scraped the sinking seeds out of the bottom of the beaker, and put those on paper towels to dry. Let them dry for probably about 48 hours. After that, just roll the paper towels a little bit. It breaks the seeds off of there, and pour them into containers. And from the seeds that we received, from the berries that we received, probably tens of thousands of seeds we have here. Each of those fruits contains dozens of seeds. And if you look at the right-hand side of the picture there, that is a single seed up against a ruler. So it's between 1 and 2 millimeters in length, the longest dimension for this. So you can see how it can go through that coarse wire mesh. But we were able to strain it out with the fine mesh. I think that's it for the depulping, right? Okay, so now.
So going into what our design was, what we actually were able to compare and kind of results in. We kind of compared the germination over a 50-cell tray, so both, whether that be the control group and the experimental group for the substrates and the sinkers versus floaters. We had a random assignment, given that this was more of a statistics project. We leaned into the idea of basic principles of experimental design. And with our random assignment, we used a simple random sample, and we assigned the 200 seeds to the experimental group, and the other rest of the 200 seeds were placed in the control group. Our controls, basically the essentials of amount of water, our lead grow lights, temperature located in the botany lab, which is pretty much around the 3-4 months that we've been germinating these seeds. And then for replication purposes, we had 50 cells in each tray with a total of 200 seeds, so 4 seeds per tray. And since they were so fine and small, I cannot confirm that we did place 4 seeds in each tray. Yeah. Oh, so by hand, we were like, yeah. So with our implementation on it, you're going to see on the left, we have our limestone mix. So we have the kind of rocky idea of it. So it looked a little more white than most of them. And then we have our controls. Some of them are controls, and then the other ones are compost mix. So it kind of goes to show that, yes, they're so small. You only see fingers going like this. So now for our results and what actually went on during these experiments.
The fun part.
All right.
Okay, so first we're going to be looking at comparing the germination proportions of seed starting soil and the clay mix. So in the top right, this is just the procedure we use. So it's something called a two sample Z interval. You don't have to worry about what that is. But basically what we found is that what this whole confidence interval means, it means that we're 95% confident that the difference between in the proportions of germination between the two groups is between negative 0.22 and 0.4. So you notice there's negative on one end and positive on one end. So we'll elaborate on that more later. And then we found that the p-value for the differences in proportion is 0.031. And so I'm also going to explain what that is once we get to the conclusion. So our confidence interval includes 0 and positive and negative values. So you can't have a positive difference and a negative difference, one or the other. And it's because it crosses through 0 like that, it indicates that there's no evidence, no conclusive evidence of a difference. And then from the p-value, what that means is if the germination proportion rates are the same, the p-value is the probability that you have the results that we got or more extreme, if that's the case. So because it was less than 0.05, but like just barely, which 0.05 is basically like where you draw the line of, is it conclusive evidence, is it not conclusive evidence? Basically, we have inconclusive evidence that the proportion of germinated cells is different in seed starting soil and clay mix. So basically, we can't say that there's a difference in germination proportions between these two groups from the statistics we did. Now for this is the mean days to germination for the same two groups. So I believe the way we did this is we were looking at first germination because we couldn't track every single of the 200 seeds in each tray. So we were just seeing which group had the first germination. Yeah. Like where you can clearly tell it had germinated. And so this group is an outlier. We'll talk about that later in the conclusion. But we found that we were confident that the difference was between negative 5.2 and negative 1.8. You see that's all negative, so there's no contradicting. And our p-value was very, very small. So basically, if the dates of germination are the same, it is almost impossible that we got the results we did or more extreme. And so because of that our confidence interval contained only negative values and that our significance test p-value was so small and underneath that threshold of 0.05, we do have conclusive evidence or convincing evidence that the mean days to germination is less for seed starting soil than it is for clay mix based on the data that we have here.
I wanted to point out this 0.05 comes from, and it comes from the opposite of the 95% assurance. So when someone tells you, "Oh, I'm 100% confident," you're 100%. Like, did you double check that, triple check that? So 95% is the rough estimate of a clear and precise conclusion that we have. So the 0.05 is the other side. That's why. So our main strategy was just to look at how the seeds were, how the plant was actually growing. Obviously, there was some accounts where it looked green, but we actually resulted that the clay mix had a lot of moss growing and a lot of mold. And so we were like, where did the white stuff come from? But they also had a lot of green. So we tried using our best. I think we used a magnifying glass at one point. But it was sure to see that, of course, there is observational error at some point, but we tried our best to see, and we did each individual, and since we knew there were only 4, if there were 5 green spots, we knew that didn't make sense.
Also to add, every group was investigated at the same time, so it's not like you looked at seed starting soil on day 15 and like, oh, it germinated, and then you don't look at clay, and then 3 years later, you're like, oh, it germinated now. You looked at them the same every time you're observing one, so it's numbers. Now for seed starting soil and commercial compost, this is actually the groups that my group did. So from our confidence interval, we have a consistent negative difference of 0.37 or to 0.11, so basically 37% to 11% difference in the proportion rates. So if you look at the graph or the table, it's a pretty sizable difference. And then our p-value similarly was very, very small, almost practically zero. And so because our confidence interval is consistent, all negative doesn't include zero, and our p-value obtained is very, very small, less than 0.05. We do have convincing evidence that the proportion of germinated cells is greater in compost mix than seed starting soil. So we're trying to probably get, well, we'll get to the applications later. And then this is the same group, but for the mean days to germination. So if you look here, they look practically the same. And that checks out with our confidence interval. It goes to negative, to positive, crosses zero, contradicts. And then our p-value is very large, 0.36. So basically saying if they are the same, the probability of getting our results, very likely. And so because of all that, we do not have convincing evidence that the mean days are any different between the two groups. And now this is seed starting soil and limestone, germination proportions. So our confidence interval was that the difference was between negative 0.30 and negative 0.8. So around in the middle, around a 15-ish, oh no, not 15, around a 20% point difference if you look at the table. So not as extreme as the previous one, but pretty considerable. And then again, our p-value was very, very small. So we know that these results aren't just due to chance. So again, so consistent interval doesn't include zero, very small p-interval. So we do have convincing evidence that the proportion of germinated cells is greater in the limestone mix versus the seed starting soil. Also, I'm going to address something real quick. So you might notice that the germination proportions and the mean days to germination are changing for the seed starting soil. The reason for that is because we had different groups to pair. So every group had seed starting soil as a control, and then they were comparing it to another group. But the numbers for seed starting soil isn't the composite of all the data. It's only from the groups that were comparing that and their respective experimental group. And then this is mean days. So the seed starting soil from the table seems to have a slight edge of around 2.5. And then our confidence interval of the difference is all positive. So it ranges from a little bit above zero to a difference of 5.3 days, which is more considerable. And then again, our p-value is close to the 0.05, but it's still less than that. So because of all that, we do have convincing evidence that the mean days to germination is less for the limestone mix. So that's good if you're trying to germinate it fast. Now for the sinkers versus floaters, it's usually generally common knowledge that or thought to be that the seeds that float are usually not good versus the ones that sink. So we were looking at that. And from the confidence interval, we did find a consistent difference, all positive, didn't go through zero. And then our p-value was quite a bit less than 0.05. So from that, we can confidently say that we have convincing evidence that the germination proportion is greater for the seeds that sink versus the seeds that float. So it seems like the general knowledge is accurate. And then this is the mean days to germination. So we found that similarly, the sinkers are better in that they appear to germinate quickly and that the floaters take longer. And again, from our confidence interval, it's consistent, all negative, and we have a very small p-value. So we do have convincing evidence that the mean days to germination is longer for the floaters and shorter for the sinkers. And lastly, we have conclusion and application. So now we're going to be switching.
Yes. So I think that was a lot of numbers. So I kind of want to recap what everything we just said about p-values and all that, because my brain is about 0.05. So what we said was when it came to clay mix and control, we saw that clay mix had not that good proportion, and it was kind of inconclusive to say how many days it actually took. When it comes, so it was kind of an X for us. We get to our compost mix, right? Our compost mix had a very good proportion difference. So compost was doing good when it came to germination, but we didn't know effectively how many days it was going to take because they almost took the same amount of time. Now, when it comes to limestone and our sinkers versus floaters, we had higher germination rates and lower mean days to germination. So that's kind of where our conclusions lie, that eventually the best compared to the seed starting soil, but individually, was the limestone mix and the sinkers versus floaters. Well, the sinkers in general, because they had fewer days to germination, so roughly almost 5 days, 7 days of difference. That's a whole week. And they had higher germination rates, almost like 20%, we said. So pretty interesting that we didn't have just one winner, we had two winners. This one.
All right, so for the errors with regards to statistics, in statistics, you always want as big a sample size as possible. There's less variance. You know the data is going to be more accurate, more realistic to what the reality is. So we had a pretty decent sample size. It's still not immensely large in the grand scheme of things. And so, for example, if you're looking at Tetrazygia in the wild, there's going to be a lot more there. So it might not be 100% applicable to that. So it'd be better to have a larger sample size. There's also things such as temperature and humidity. So you can put like the AC in the room, but there's still the external world that's going to affect the humidity and the temperature of the air inside the room. And then that can obviously impact the rates of germination as well as the days to germination. And lastly, there's also human errors such as maybe inconsistently measuring, like collecting data. So maybe they didn't keep a consistent schedule with how often they were doing it, or they did something else inconsistently, or perhaps not the best way. They might have not measured perfectly accurate. Like you might have 200 seeds, but you accidentally put 204, or you miscount how many germinate, or you miss. Sorry.
Some names comes to mind. So apart from us doing algebra wrong, we might have accidentally looked at it wrong. And something known as observer bias is when we document unintentional things because of our creative minds that we have. We say, oh, it germinated, right? Woohoo! So, and again, that goes back to our first day of germination. So we all, since we had 3 different periods that we saw each other on different days, we had on Mondays and Tuesdays, we didn't exactly know what was the first day of germination in total. And since most of the seeds were planted at different rates, watered, they had an average amount of water, but some people that go, and then some people that are like, so it kind of affects the capacity of.
We call that a confounding variable.
Yes.
Okay, so we have a lot of urgency with maintaining native plants in South Florida. I think I heard recently that there's more people trying to develop into sort of native plant areas, and that's kind of been the trend over the last, ever since like the 1920s when South Florida started being developed, increasingly encroaching on the native lands and native plants, and they're becoming increasingly threatened or even endangered. And so obviously we want to figure out the best ways to take care of our plants and keep them safe. So it's important to understand which medias are best for helping scientists and other groups increase the amount of Tetrazygia bicolor and other plants in the wild, as well as in captivity to perhaps study them further. And then with regard to the limitations of our research, so the concentration of the substrate mixes. So we got particular, like might get from a certain brand seed starting soil, and you get a certain compost mix, but not all compost mix and limestone mixes are going to be exactly the same. So that's another thing to consider. like a limestone mix that we get might not be exactly the same as one in Mother Nature. So that could be something that skews the results.
And we did 50-50 on our germination for experimental group and a control group. So what would have happened if we had 75% of it concentrated on experimental and 25% on control? The difference is that we have.
We had the same number of seeds and cells for each group. We didn't have more for, say, the experimental than the control group.
So the often question is, okay, what do I do with this? Thank you for the data, but what can we imply with all of this? And it goes to show that identifying that our sinkers with their high germination proportions and their low mean days and the limestone group kind of, they intertwine together because it's perfect. You know, when you are propagating depulping the seeds and everything, and you're saying, well, I can use the sinkers and I can use the limestone substrate, and boom, double the results. So it just helps to show that the large amounts that we do and the more, obviously the bigger the sample size, it can have a meaningful impact and it can help restore this habitat that is so imperiled and endangered in less than 1% at this point. And so doing this at a much larger scale and with different ideas in mind, like improving their concentration, doing them and even testing them like against each other, a limestone group compared to a clay mix, not just compared to a control. See how that goes, and different brands. So keeping it consistent, I think for every experiment, every time you repeat it, it gets better and better. And so I think repeating that at a larger scale with more plants in mind, more seeds, can definitely turn out to be pretty accurate. And so we come to the end, and I don't want to say goodbye yet, but here's just an overview of our bibliography. We used a lot of stuff from the Institute of Regional Conservancy, from Fairchild, from FNPS themselves, and not a Wikipedia page. So it kind of, every website, every kind of article that we saw kind of had the same idea that yes, it's imperiled, but there is hope, right? Because even though for Brian it didn't work out, for us it did. And it's gonna help, and we're gonna have a lot of with our new Myconia bicolor, I guess. So onto our closing remarks. We want to thank Fairchild Tropical Botanical Gardens Conservation Team. I don't remember all the names you just said. Lydia, I think, was one of them. Jen, Brian. And so we also thank the Date chapter of the Florida Native Plant Society for giving us this opportunity to present to all to you today. I think we have a lovely crowd. My number one fan. Yes. And then my special thank you goes to the one person that really made this happen, that bugged us to get this presentation done. Mr. Kearns and our AP Statistics and our Botany program at Jose Marti. It really is incredible. I'm in a bunch of extracurriculars that I see other schools in the Hialeah Gardens area. I visit Miami Lakes Educational Center. I've been to Matter, and they're like, we don't have this forest growing in our backyard. We have 3 community gardens, and that's about it. So the work we do really goes to show that we're such a small school, like middle and high school, and we're 800 kids. So, so thank you, Mr. Chris.
So there's one more slide.
Yes, there is one more slide. Oh, I mean, we could talk about the that I don't know what day.
Oh, this was 50 of the plants were used in a restoration project at the Nixon Smiley Preserve. I was there. I tried to get some of our students to come. It was a bad weekend for that. I'm glad they didn't come because it's for hours and lightning and you know, Scott, you're out there, and Brian, you're out there, and yeah, I was on my hands and knees planting stuff for about 45 minutes. That's about all the work we got in before the rain. But 50 of the Tetrazygia are out there, and I think after that rain, it didn't rain until last week. So I'm hopeful that they're surviving. But you know, something that I have learned from you, Brian, is hey, once you let go of the plants, you let go of the plants. You know, and then you just you hope the best for them.
You're sending the kids off to college.
Yeah, that's right. That's right. Very good. So before, and these are the 5 students here that presented. That's Chris on the left, Celine, there's Nestor, you recognize him in the middle, there's Alejandro next to him, and that's Nicholas on the right-hand side there. Something that I do want to mention though, and both Nestor and Alejandro alluded to this, is that there were multiple groups that were doing these experiments between 3 different periods. And the reason I did that was so that we would have a sufficient sample size with combined data to be able to assume normality, to be able to do inference procedures for both proportions and for means. Because in previous years, you know, I might have 10 groups and each of them doing their own experiment. But unless things worked out perfectly, they didn't have sample sizes that were appropriate, that where you could assume normality and then apply the statistical inference procedures that we did. So I got smart last week and duplicated experiments from period to period. So that's why the sample sizes were 150 or 100, because those were multiple periods involved. And again, that's one of the reasons why we had some confounding variables. Because we had different people doing different experiments with different habits, different observational skills,
Different watering skills.
different motivations. I mean, some kids were less motivated. They didn't get much in preparing presentations, but you know, I thank Alejandro and Nestor for not just doing the work last year, but doing the work this year, because you know they're not in this year. But at the beginning of the year, I asked, hey, would you like to make a presentation that compiles all our research from last year? And I'd like to be able to not only present it here, but to in other settings as well. So hopefully we'll get that opportunity. Hopefully Celine and Chris and Nicholas will get a chance. So with that said,